This video covers the lymphatic system, including tissue fluid and the immune response. The lymphatic system functions to drain excess tissue fluid that doesn't return to the bloodstream and initiate the immune response to eliminate pathogens. Major structures include lymphatic vessels, lymph nodes, the spleen, and the thymus. Lymphatic structures are divided into lymphatic vessels and lymphoid tissues and organs. Primary or central organs are the bone marrow and thymus. Secondary or peripheral organs include lymph nodes, the spleen, and mucosal tissues. Lymphatic vessels are similar in structure to veins, being thin and having valves. However, they are a one-way street, originating as capillaries in the tissues and ending at the veins. Lymph is the excess tissue fluid that enters the lymphatic vessels. Its functions include transporting proteins and other molecules to the bloodstream and moving foreign particles to lymph nodes. The thymus is located behind the sternum and is larger in children. It is the site of T-cell maturation after they migrate from the bone marrow. Lymph nodes are located along lymphatic pathways, except in the central nervous system. They filter out potentially harmful particles and are responsible for immune surveillance. The spleen is located in the upper abdominal cavity and is similar to a lymph node. The red pulp disposes of red blood cells, while the white pulp houses lymphocytes and is the site of antigen presentation. Mucosa-associated lymphatic tissue, or MALT, is lymphatic tissue in the mucosal membranes throughout the body. It contains T cells, B cells, and macrophages, and can encounter antigens passing through the mucous membranes. MALT is found in several locations, including the lungs, digestive tract, nasopharynx, skin, breast, and salivary glands. Cells of the immune system are broadly grouped into myeloid cells, mostly involved in the innate immune system, and lymphoid cells, which are part of the adaptive immune system. Myeloid cells include macrophages, which engulf and kill invaders, induce inflammation, and activate other immune cells. Mast cells play a role in allergic responses and respond to parasitic worms. Granulocytes include neutrophils, which are the most numerous and are phagocytic, playing a role in bacterial infections. Eosinophils and basophils defend against parasites and contribute to allergic responses. Dendritic cells take up and degrade pathogens. Their main function is antigen presentation, activating cells of adaptive immunity, thus bridging the innate and adaptive responses. Lymphoid cells include natural killer cells, which come from the lymphoid lineage but are not antigen-specific. They recognize and kill abnormal cells, like tumor cells and some viruses, keeping them at bay until the adaptive response is active. B lymphocytes, or B cells, have antigen-specific receptors called BCRs. Once activated, they divide to form plasma cells, which produce antibodies, the secreted form of BCRs. T lymphocytes, or T cells, are categorized as helper T cells, which initiate responses from other cells, cytotoxic T cells, which kill infected cells, and regulatory T cells, which suppress and control the immune response. The immune system involves cells and biochemicals for surveillance and destruction of non-self entities. The innate response is quick and non-specific, while the adaptive response is slower and specific. Cytokines and chemokines are chemical signaling molecules. Cytokines are secreted proteins that affect the behavior of nearby cells, acting as pro-inflammatory, regulatory, or growth factors. Chemokines attract cells to an area through a density gradient. The innate immune response involves macrophages recognizing and engulfing pathogens through receptors. They present antigens on their surface and secrete cytokines and chemokines. Inflammation, characterized by heat, redness, swelling, and pain, recruits proteins in cells like macrophages and neutrophils to infected areas. It increases lymph flow, bringing microbes and APCs to lymphoid tissues, and later recruits effectors of adaptive immunity. Pathogens can trigger the activation of complement proteins. This activation initiates a cascade that destroys the pathogen and can also promote inflammation. Innate immune recognition involves pattern recognition receptors, or PRRs, on cells like macrophages, neutrophils, and dendritic cells. These receptors recognize PAMPs, or pathogen-associated molecular patterns, which are present on microorganisms but not on body cells. The innate response initiates the adaptive response. Macrophages engulf pathogens and present antigens to lymphocytes. Dendritic cells are triggered to activate T cells. Adjuvants, like bacterial extracts and vaccines, initiate an innate response, which then activates the adaptive response, leading to successful vaccination. The adaptive immune response is initiated by antigen-presenting cells, primarily dendritic cells and macrophages, which engulf and degrade pathogens, then move the antigen to the cell's surface. Dendritic cells also have costimulatory molecules that stimulate T cells. This process occurs in secondary lymphoid tissues. Upon activation, naive lymphocytes become lymphoblasts and undergo clonal expansion, dividing rapidly to produce about 1,000 cells specific to the target antigen. These cells become effector cells, such as plasma cells for B cells and cytotoxic or helper T cells for T cells. Most cells from clonal expansion die, but some T and B cells remain as memory cells. These memory cells are quickly reactivated upon re-encountering the same antigen, providing long-lasting immunity. Mechanisms of adaptive immunity include the cell-mediated immune response, where T cells destroy intracellular invaders, and humoral immunity, where antibodies detect pathogens in blood and extracellular fluid. Other mechanisms are similar to the innate response, involving macrophages, neutrophils, and complement. In T cell and cell-mediated immunity, CD4 cells are helper T cells, and CD8 cells are cytotoxic T cells. They recognize peptides on MHC molecules, which are displayed on the surface of cells. MHC class I molecules display proteins synthesized in the cytosol, including viral proteins, and are recognized by cytotoxic T cells. MHC class II molecules are expressed by APCs and display proteins derived from phagocytosis or endocytosis. They activate CD4 cells. Stimulated effector cells secrete effector molecules, mainly cytokines, which recruit other effectors. CD8 cytotoxic T cells directly kill infected cells, while CD4 helper T cells can differentiate into different types of T cells. Th1 cells help control bacteria in macrophage vesicles. Th2 cells promote responses at mucosal surfaces, particularly against parasitic infections. Th17 cells promote responses with neutrophils against extracellular bacteria and fungi. TFH cells aid in B-cell activation within lymphoid follicles. Antibodies are secreted B-cell receptors with two variable regions for antigen binding and a constant region. The constant region determines the effector function and how the antibody interacts with immune cells to dispose of the antigen. Antibody actions include neutralization, which blocks pathogen access to cells, opsonization, which coats pathogens for phagocytosis, and complement activation, where constant regions activate the first complement protein, enabling phagocytosis. Antibodies are found in plasma and extracellular fluid. Antibody-mediated immunity is called humoral immunity, as body fluids were historically known as humors. The primary immune response is the first activation of T and B cells, leaving behind some memory cells. The secondary immune response occurs when the antigen is encountered again, allowing memory cells to mount a more rapid and long-lasting attack. Immunity is classified as naturally acquired immunity, occurring after exposure to the antigen, artificially acquired immunity through vaccines, artificially acquired passive immunity via injection of antibodies, and naturally acquired passive immunity through antibodies passed from mother to fetus. The first vaccine was developed in 1796 using the cowpox virus against smallpox. Vaccines mimic a natural infection, allowing the immune system to respond normally and leave behind a supply of memory T and B cells. Vaccination is the act of giving vaccines to prevent disease. Immunization is the resulting acquisition of protection against a disease. Successful vaccination results in immunization. Live attenuated vaccines contain weakened, live microbes that no longer cause disease, eliciting a response involving memory T and B cells. Drawbacks include the need for refrigeration and a risk of mutation. Inactivated or killed vaccines are the most common type today. They contain viral pieces killed with heat, chemicals, or radiation. They stimulate B cells to produce antibodies but are less potent and require booster shots. Subunit vaccines contain only the parts of microbes, mainly antigens, that stimulate the immune system, often causing fewer adverse reactions. Toxoid vaccines use inactivated toxins, called toxoids, to combat the toxins produced by pathogens, stimulating antibody production.